light energy into chemical energy. What's the form in which this chemical energy is moved from the light-dependent reactions to the Calvin cycle? What are the molecules here? You guys remember the energy storage molecules?
ATP.
Yeah, good. And what was the other one? Yeah, NADPH. So last time I showed you guys the Z scheme. The Z scheme depicts the electron transport chain that's necessary to convert this light energy into this chemical energy. So you can think, one way to think about it, one way that I thought about it when I was in undergrad, is to think about this as kind of a way of charging up the chemical energy in the chloroplast by using the sunlight so that you can power the reactions over here. This is where the energy is going to be spent. And the important thing about this, what's the input to the light-independent reactions of the Calvin cycle? Yeah, carbon dioxide. So what's important here, this is the carbon capture phase. This is where you're taking carbon out of the atmosphere and using it to build that plant biomass that we talked about. And the product is sugar or carbohydrates that can be built out of those sugars. We also talked about 3 phases to these light-independent reactions. The 3 phases I want you guys to know are carbon fixation, which just refers to carbon capture. You're taking gaseous CO2 and putting it into organic molecules that can be processed by the reactions of the Calvin cycle. And the second is reduction. Reduction is where you're taking some of that ATP that was produced over here in the light reactions and using it to increase the amount of chemical energy in these organic molecules that are produced through carbon fixation. So we're converting 3-PGA to G3P molecules. And these are the high energy building blocks of sugar molecules. These are the high energy building blocks of sugar molecules. But not all of these G3P molecules are used to synthesize sugar. What else does the Calvin cycle have to do to make sure photosynthesis can proceed indefinitely? Anyone remember? I haven't put this on the board yet, so I'm really asking you to dig deep. Yeah, Sandra.
Does it have to make NAD plus?
Not NAD plus. Remember, that's being produced over. Oh, sorry. Well, some of the NADPH is used for this process, and so the product of that is NAD plus, which goes back over here to get repolished. You guys remember what goes into the initial carbon capture phase? What is CO2 bound to, and what mediates that carbon capture reaction?
So with Rubisco?
Rubisco is the enzyme. Right? Carboxylases the carbon dioxide that captures the carbon. And what is what's the other reagent in this reaction? The other reagent is this 5-carbon RUBP molecule. So RUBP plus CO2 is a total of 6 carbons. That reaction gives you 2 3-PGA molecules. And then those can then be converted into G3P and ultimately to sugar. So the other thing that has to happen is some of these G3P molecules are used to regenerate the RUBP necessary for the carbon capture phase, for phase 1. So you have to regenerate RUBP for that carbon capture reaction. Does that feel like a refresher? This initial carbon capture phase is the key to the difference between C3 and C4 plants. Display. Chicken. I ended. Oh, there we go. Now we're in business. Okay. So we ended last time introducing this graph. Let's once again remind ourselves, what's the way to walk through a graph? Let's start with the axes. This is called the light response curve. What we're seeing is that as the amount of incoming light increases, the amount of photogenically active photons increases per unit area, per unit time, from low to high, we see a response in the rate at which carbon dioxide is captured or assimilated by the Calvin cycle, by the whole photosynthesis process. So the curve shows that as you increase the amount of light, the rate of carbon dioxide assimilation increases until what happens? Until what happens? Does it increase forever?
No, it'll max out.
So what happens? It maxes out. It becomes light saturated. So something else other than available light is limiting the amount of carbon dioxide that can be assimilated. In this region, the reaction is limited by the amount of light. If you increase the light, you get more assimilation. Now here, if you increase the light, and there's very little increase in carbon dioxide assimilation. Is that a question in the back? You okay? Okay, good. So there's a few important points now that we've talked about the axes and the general shape of this graph. Down here is this intercept here is called the dark respiration rate. Does anyone remember why it's called the dark respiration rate? Yeah, it's the rate at which CO2 is lost, not taken in, but lost to the atmosphere due to respiration in the dark. So what do you think this intercept is here, where the curve intercepts the x-axis? What does this mean? What's balancing out at that point? Remind yourself, where is photosynthesis taking place in the plant? In what organelle? And what organelle is CO2 being evolved? Is CO2 being produced in? Mitochondria. So if you got both of these organelles doing both of these processes at the same time, what is this? What's the significance of this light level, this low light level right there? Say it a little louder. It's sort of an equilibrium between the two. It's not necessarily a stable one, but it's a balance here where there's just enough light to assimilate just enough CO2 to offset kind of the normal operating costs of cellular respiration for the plant. So this is where the amount of incoming light compensates, or the amount of incoming light powers enough photosynthesis to compensate for the normal respiration rate. So that's your light compensation. Okay, and again up here, as we already said, this is where something else other than light starts to limit the rate of photosynthesis. What do you guys think might limit photosynthesis in that region, in that plateau region? Any thoughts? Tony, give me one of them.
Probably water. If
So,
there's not enough
water
water.
is usually a first guess, and it's a reasonable one because it's one of the other inputs. So if you're not limited by light, maybe something else. In fact, most of the water that plants lose just gets transpired into the atmosphere. So if you had so little water that that was what was limiting the light reactions, the plant would have dried out and died a long time ago. So it turns out it's not water that's limiting that part. Someone else want to, thank you for playing, someone else want to try? CO2. CO2 could be what's going on. We're going to talk about a curve for CO2, but generally, what do you think the concentration of CO2 inside the leaf is going to be relative to the concentration of CO2 in the atmosphere? Would you guess that it tends to be lower in the leaf or higher in the leaf? Lower. Why lower in the leaf?
I don't know.
Because it's being used.
It's being used up. It's being consumed by the Calvin cycle. And so relative to the external environment, you have a gradient that's moving CO2 into the plant. So it turns out, usually, it's not CO2 either. Can you think of anything else that might limit photosynthesis at those bright conditions? Yeah.
Space that it diffuse reactions?
Space is kind of a good guess, but it's actually the rate of these reactions, so it becomes limiting. It's how much Rubisco do you have available here to fix that carbon, and how quickly can those 3PG molecules be metabolized into G3P, into sugar, into RUBP, and keep the cycle going. So even if you got plenty of this stuff, how fast can these reactions go? That turns out to be the thing that can limit. The capacity of Rubisco to capture carbon dioxide and go through that metabolism works okay as long as the concentration of CO2 inside the leaf is sufficiently high that it can grab those CO2 molecules and use them in that carbon capture reaction. Let's see. So this is the anatomy of a leaf. My diagram here is a little bit funny, but even though leaves seem quite thin, whether you're thinking about a grass leaf, a Conza prairie, or one of these walnut leaves right outside here, whatever they are, it seems quite thin, but there's actually anatomy in it. There's structure in the leaf, and it's relevant here. So if you imagine cutting a leaf and looking at it inside with a microscope, what you'll see is a bottom layer and a top layer here. That's called the epidermis. And then the middle part in between those two epidermal layers is called the leaf mesophyll. It means middle leaf, mesophyll, middle leaf. And that has some zonation too. Usually, the lower part of the mesophyll is kind of spongy, at least there's lots of air gaps, air spaces in between the cells. That's what we mean by spongy. And in the surface of the epidermis, adjacent to that spongy mesophyll, are these pores called stomata. Most of the leaf epidermis is covered in a waxy cuticle that prevents the movement of water across that epidermal layer. Therefore, the water that does move across the epidermis has to go through these stomatal pores. That's also how the leaf gets its carbon dioxide, by opening those stomatal pores. So if you open your stomatal pores to take in all that carbon dioxide, what do you run the risk of doing, given what you see in this diagram here? What's the cost or the effect of keeping your stomata open all the time to capture that carbon dioxide? Your CO2 molecules diffusing in, because the CO2 concentration is lower in the leaf than outside, so they're diffusing in. As long as those stomata are open, what else is happening?
Water getting.
Water actually goes out, because inside the leaf, where you have these moist tissues, the relative humidity in there on the surface of the cells is basically 100%. All the cells are wet. So the air spaces between them have really high humidity. Whereas out here in the atmosphere, humidity is lower, so water diffuses out. So as long as you've got your stomata open to take in CO2, you're losing water. And that's why I said earlier, most of the water a plant uses and takes up from the soil is just lost through the stomata. It's not used for light reactions, only a small fraction. is used for light reactions. So that's great. If you keep your stomata open, you get CO2 going in, and you can power the Calvin cycle, or you can fuel the Calvin cycle. Excuse me. But the cost is losing water. What happens to a plant that loses too much water? It can die, but what happens before your plant dies? What happens if you get to it just in time, right? It's wilt, right? Yeah, so if the vacuoles in the plant cell lose too much water, the rigidity of the plant cells decreases because it uses that water pressure to feel crisp. And as the water is lost, it becomes kind of soft and wilty, and the plant eventually can die. So, this is the critical part here, right? CO2 uptake is driven by a concentration gradient, which we talked about. And it turns out the concentration gradient of carbon dioxide, or the concentration of carbon dioxide in the atmosphere today, is a little over 400 parts per million. Now that varies. The 400 parts per million is upper atmosphere, like if you measure CO2 on the island of Hawaii. In a city like Kansas City, during the day, if you're downtown, it could easily be 800 parts per million locally. But generally, we've got CO2 somewhere between 400 and 500 parts per million in the atmosphere, and it's lower in the leaf, so it moves in. Let's think about another curve you might see on an exam. Here now we have the concentration of carbon dioxide in the cell, in the leaf mesophyll. What's the concentration of carbon dioxide in the leaf mesophyll? Well, it's probably going to be somewhere around 400 or lower, because that's about what the atmosphere is, and you know that it's lower in the leaf, somewhere down here. But it could go higher if you added CO2 to your greenhouse or something. And then on the y-axis, we have the rate at which that carbon dioxide is assimilated from the gas in the leaf mesophyll into the Calvin cycle, into sugars. And the curve looks somewhat similar. Here, as long as CO2 concentrations are low in the leaf, the rate of photosynthesis is limited by the availability of carbon dioxide. More CO2, more carbon assimilation. And eventually, once again, it starts to what? What happens up there?
CO2 saturated.
A little louder?
CO2 saturated.
Yeah, it becomes CO2 saturated or limited by something other than the availability of carbon dioxide. Once again, here I've got rate limitation is by something like the activity of rubisco or metabolism in the Calvin cycle. So leaves spend most of their time down here. No, I just asked you this question. So why aren't stomata always open? It's because they'll lose too much water. So this brings us to the concept of water use efficiency. So when these stomata are open, the plant is getting carbon dioxide and it's losing water, and the ratio basically of carbon dioxide gain to water loss is what we mean by water use efficiency. It's the ratio between the gain of carbon dioxide in photosynthesis and the loss of water due to transpiration. Transpiration is just a special word we mean for the loss of water through plant leaves, through photosynthetic tissue. What I want to emphasize here is that having low or high water use efficiency isn't necessarily a good or a bad thing. It just influences how well the plant performs under different conditions. If there's plenty of water, you don't have to conserve it. Dump all the water into the atmosphere and keep those stomata open and grow fast. That's great. But if the conditions become water limiting, then having higher water use efficiency might be good if you're thinking about carbon gain. So, water use efficiency for any given plant depends on diffusion. Specifically, the differences between the vapor pressure in the leaf and in the atmosphere, 100% to some humidity level in the atmosphere, and the ratio of the concentration of carbon dioxide in the atmosphere to the internal carbon dioxide concentration of the leaf. That's what's going to drive kind of the strength of those two arrows that are going in and going out. So can you think of something that a plant could do or might experience that would increase its water use efficiency? if you're losing water as you gain carbon dioxide, what could increase your water use efficiency? Anyone who hasn't spoken up yet today, take a stab at it. You want to lose less water per unit carbon gain. What would you do as a plant? Can you think of a way that you might manipulate the amount of water lost through the stomata? You could close them. Yeah, you could close the stomata. Another thing that could happen is if the concentration of carbon dioxide in the atmosphere goes up, that would increase the ratio of carbon gain to water loss. But the main thing a plant can do is change the rate at which water is lost through opening or closing the stomata. But what's the challenge that comes with closing your stomata? Yeah, then you're not getting that CO2. There's another problem too. Does anyone remember? What the O stands for in Rubisco. It's a carboxylase enzyme. It fixes carbon. That's what the C stands for. What's the O stand for? It's an oxygenase also. So some fraction of the time, when the Rubisco has the available RUDP and available carbon dioxide, instead of binding that RUDP to the carbon dioxide and give you those 3 PGA molecules, it'll bind the Rubisco to oxygen. That gives you a 2-carbon molecule and a 3-carbon molecule. And so you don't get the right ratio to power, to fuel the Calvin cycle and produce those sugars. So when that happens, that's called photorespiration. So far we've only talked about the Calvin cycle, which is right here. RUDP and CO2 go into Rubisco, and you end up with these 3 carbon molecules. Energy gets added to make sugars and to regenerate RUDP. If instead the RUDP reacts with oxygen mediated by the Rubisco enzyme, CO2 is produced instead of sugars. You get some PGA that can be used for regeneration of RUDP, but this costs energy, just like the Calvin cycle does, and you don't get sugars out of it. So it's a problem for the plant. And what we're going to see is that under certain conditions, when this becomes really problematic, we see selection favoring the evolution of C4 photosynthesis multiple times. So the energy that is used up here is used to just regenerate that RUDP and be ready for a normal flow of the Calvin cycle. And this is what we're going to see that C4 plants overcome. Here's another way of looking at this. We've got RUDP, that 5-carbon molecule, plus oxygen instead of carbon dioxide, gives you 3 PGA, a good thing, but also this 2-carbon molecule here, which the plant can't use for synthesis of sugars. Oh, sorry, here we go. So then the point here is that the risk of photorespiration is highest when the internal concentration of carbon dioxide in the leaf is low.
I think Kenmore has a question.
Yeah.
Is that right?
Does photorespiration occur when there's not enough carbon dioxide?
Yes, so that's exactly where we're going. Yeah, when there's not enough carbon dioxide, but it's inside the leaf, that's when the risk of photorespiration increases. So Rubisco can work with either carbon dioxide or oxygen. It favors carbon dioxide. So under normal conditions, when there's plenty of water, the stomata are open, the risk of photorespiration is really low. But when you close those stomata to conserve water, and the carbon dioxide concentration in the leaf goes down, down, down as it gets used up by the Calvin cycle, then there's all these other oxygen molecules just in the leaf mesophyll, and those start getting used up in photorespiration. A good question, thank you. So if closing the stomata can increase the rate of photorespiration, under what environmental conditions do we expect the rate of photorespiration to be highest? Here's a 3D graph to read. So once again, let's walk through the axes. So we've got temperature from low to high, so cool versus warm. We've got the concentration of carbon dioxide in the leaf, actually in the chloroplast where it's being used, from low to high CO2 concentrations. So down here in this front corner, you've got high CO2 and low temperature. And then our y-axis is the ratio of photorespiration to the production of sugar, basically the photosynthetic rate, carbon assimilation. So photorespiration to photosynthesis ratio. So where is the rate of photorespiration highest relative to normal photosynthesis on this graph? What combination of CO2 conditions and temperature conditions? Yeah, so warm, and what about the CO2 conditions? High or low? Yeah, low. So when CO2 is low and temperature is high, that's when plants tend to have warm conditions, so low humidity, dry soils, these kinds of things. That's where you're going to get environmental conditions that favor C4 plants. Whereas if CO2 concentrations are high, where the temperature is low and the plant has access to lots of water, the base efficiency is going to be really low, and a plant can, using normal C3 that we've talked about so far, fix all kinds of carbon, and the risk of photorespiration is low, so photosynthesis is high. So these dots here represent the internal CO2 concentration of a typical C3 plant in a world with about 400 parts per million. So you can see that as the temperature changes, the ratio of C3 to C4 plants can change. That'll make more sense in a moment. So, how do plants overcome the limitations of photorespiration and water loss that are imposed on them by having to regulate only their stomata opening and closing? That's the question. And the solution is a carbon concentrating mechanism that avoids the risk of Rubisco fixing oxygen. It concentrates the carbon dioxide so that Rubisco is only exposed to the carbon dioxide, and the risk of photorespiration is thus low. A moment ago, I told you about leaf anatomy. Here's another section of a leaf. We've got an epidermal layer down here. We've got an epidermal layer up here. We've got a leaf mesophyll. All the little cells in here are leaf mesophyll cells. And this sort of clump right here, this clump right here, those are the veins. Those are the conduits in the leaf that are delivering water, distributing water throughout the whole leaf, so that all those mesophyll cells stay wet and water can be transpired. And in a normal C3 plant, you have a spongy mesophyll with those veins in it, and then a palisade mesophyll up here, which just has a high concentration of chloroplasts. But this mesophyll is what matters here. In a C4 plant, the anatomy looks a little bit different. These are the veins. Do you notice anything funny about these veins compared to those veins? Can you describe what you see? How are these veins different than these veins? I'm just asking about like shapes and sizes. Say it again? Describe what's different about these and those.
Round. There's a bunch of little ones and the big ones.
Yeah, so Caden sees these big round cells here surrounding little cells. Here, we don't see those big round cells surrounding the veins, do you? These special cells here are called bundle sheath cells, And this pattern that you're seeing is called Kranz anatomy, K-R-A-N-Z. These bundle sheath cells are where carbon is concentrated and oxygen is excluded. So that's where the Calvin cycle takes place. And this is a grass leaf, and this is a typical C3 form leaf. Here's a diagrammatic version of the same thing. Both leaves have normal mesophyll cells out here, and that's what this box represents. Only the C4 plant has these bundle sheath cells surrounding the vein. In a typical C3 plant, CO2 and ribulose bisphosphate generate 3-PGA, right? Generate that 3-carbon molecule. In the C4 plant, these mesophyll cells out here, away from the veins, capture the carbon with a different enzyme, an enzyme that doesn't fix oxygen. This enzyme is called PEP carboxylase, PEP carboxylase. It's not an oxygenase, it's just Pep C. And when this organic molecule here is fixed to CO2 by PEP carboxylase, this phosphophenol, I didn't say because you guys don't have to worry about these names, but what I want you to know is that the product of carbon fixation by Pep C is a 4-carbon molecule, not a 3-carbon molecule. That's why they're called C4 plants. The initial product of carbon capture in phase 1 of the initial product of carbon capture by Pep C is a C4 molecule. That's why they're called C4. Then the plant actively pumps this C4 molecule in the form of malate into the bundle sheath cells. When I say active, I mean it costs energy. It spends ATP to move that malate into the bundle sheath cell. There, the malate is broken down to release the carbon for the rubisco enzyme to use it to power the Calvin cycle. So in this environment, you've got an oxygen-free or low oxygen environment where the risk of what is low? The risk of photorespiration is low. So that's the carbon concentration mechanism. It's actively pumping the malate into the bundle sheath cells where it can be used for the Calvin cycle. So we've got a spatial separation of the initial carbon capture and the Calvin cycle. The nice thing is, at least in grasses, most C4 plants, you can see it. You can see the Kranz anatomy. These bundle sheath cells are really distinctive, and the sugar that they produce at the end of the Calvin cycle can be loaded right into the flow of the leaf veins and transported down to the roots or the growing meristems or wherever that sugar needs to be used for growth, for cellular respiration. This is a paradermal section of a leaf. You're looking down at the surface of a grass leaf. These little spiral areas look like little train tracks. These are the veins that are transporting water, and these big round brick-like cells that are surrounding those veins are the bundle sheath cells. So imagine zooming way in on a grass leaf. Those are your veins, and these are your bundle sheath cells, where that rubisco is sequestered, and photosynthesis proceeds without photorespiration. So kind of recapping on that with some text for you guys. How do bundle sheath cells work? We have spatial separation of initial carbon dioxide capture and carbon fixation between the mesophyll cells and the bundle sheath cells. The enzyme that initially catalyzes the capture of CO2 is this PEP carboxylase, and it results in a 4-carbon acid. This 4-carbon compound is actively transported into the bundle sheath cells as malate, where it can be decarboxylated to release that carbon, and the Calvin cycle can proceed without oxygen. So here you've got a simple diagram showing the difference between C3 and C4. Here's a slightly more complicated, or intricate version of that, but what it shows is that this process right here is active. This is what requires energy, ATP. So it's great to be a C4 plant because you can reduce the risk of photorespiration, but there's a cost associated with that. Why do you think malate has to be actively transported from the from the mesophyll cells into the bundle sheath cells. Any guesses as to why that has to be active, why that takes energy? Yeah.
Does it have a higher concentration or something?
Yeah. You can have a higher concentration of malic acid here that can be, and the breakdown of malic acid or malate can be rate limited by all this metabolism. So you have to pump against the concentration gradient, and that's what requires energy. So we sort of talked about this already. Concentrating malate and producing PEP carboxylase, synthesizing that extra enzyme, has energy costs. So under what context might this be beneficial, of doing all this extra C4 stuff? Under what conditions would spending that extra energy on synthesizing PEP carboxylase and pumping malate and all the other things that come with being a C4 plant, under what conditions might it be worth it?
Prairie.
A prairie. Good. Why a prairie?
Because it's bright and sunny.
So it's bright, so you're not going to be light limited. And what else do you guys know about water availability in the prairie compared to other places that maybe have forests? Think about the difference between water availability on average over the course of a year between a forest and the prairie. Remember that gradient as we move from east to west across the continent? We go from forest to prairie to desert. What's water availability going to be like in the prairie? Increase compared to what?
You said decrease.
Oh, decrease, yes. So lower, water availability is certainly lower than in a forest. Yeah. So it's worth it if there's plenty of sunlight energy and water can be scarce, at least seasonally. It's also the case that we have relatively low carbon dioxide concentrations in the atmosphere today compared to the general history of Earth. C4 photosynthesis has evolved many times in different groups of grasses and in a few groups of forbs as well. Many times, and almost all of that has been in the last 30 or so million years. And C4 dominated ecosystems like grasslands are no more than about 10 million years old. So this kind of a world where we got C4 dominated grasslands is a new kind of ecosystem, a new kind of biome. And that's because having low carbon dioxide concentrations in the atmosphere, and therefore low CO2 concentrations in the leaf, and high photorespiration rate or risk, that's all a new thing. So this is a new solution to conditions where it's warm and carbon dioxide concentrations are low. So this graph shows you the growing season temperature from cool to warm and carbon dioxide concentration in the atmosphere from low to high. And we're right about in here at 400. And it's been lower in recent history. It's been closer to 300. So warm places, hot places where the soils can be dry and humidity is low, and also experiencing low CO2 are going to favor C4 plants. Whereas if it's cooler and CO2 is reasonably high, C3 are going to be favored. Here's another way to look at that same effect here. Now we've got our carbon dioxide concentration on the x-axis. We've got our rate of photosynthesis or carbon assimilation on the y-axis here. And notice that with a C3 plant, the intercept with the y-axis here is lower. It's going to be further down. That's because of those increased energy costs, right? So when you've got low CO2 concentrations, sorry, I said the wrong one. C3 has a lower intercept here. So when you've got lower CO2 concentrations, which curve is higher, the C3 curve or the C4 curve? to read the graph. Which curve is higher at low CO2 concentrations? Yeah, the C4 is higher. So you get more primary production as a C4 plant when CO2 is limited. When CO2 concentrations are low, then you use a C3 plant. But as you increase CO2 concentrations, what happens to the risk of photorespiration? What happens to the risk of photorespiration as CO2 goes up? Think about rubisco. It can use carbon dioxide or oxygen, prefers carbon dioxide, but it will use oxygen if CO2 is low. So if you increase CO2, what happens to the risk of that oxygen use? It decreases. So the energy costs associated with being a C4 plant become unnecessary, and the C3 plant can start to win and start to have higher productivity, higher photosynthesis rate. So there's that switch somewhere in here where one has the advantage versus the other. And we can see this switch about who has the advantage spatially from the satellites. So here's a map of the prairie region, the prairie biome of the United States, going from Texas down here. You can see Texas all the way up to Montana over here. Right here is where I saw that bison carcass I showed you a couple weeks ago in Teddy Roosevelt National Park. We talked about the water availability gradient going east to west. Someone remind me what the gradient is going from north to south? So the main water gradient is east to west. What other environmental parameter or environmental variable varies north to south besides water? Yeah, temperature, right? So we've got warm, wet ecosystems down here. We've got warm, dry ecosystems here, cooler and drier, and cooler and wetter. This is complicated a little bit by growing season, but what I want you to see generally is that the C4 plants tend to dominate in the warmer regions, and in the cooler regions we tend to get C3 plants, especially in the early cool growing season. And then we've got tall grass prairie, mixed grass prairie, and short grass prairie. Remember that? So we've got our tall grass C4, there's Ponza Prairie in the Flint Hills region. Here's our short grass C4 prairies in West Texas. Short grass C3, more tall grass mixed. Seem okay? Can you see the relationship between water availability and temperature here and their influence on photosynthesis and the relative advantage of C3 versus C4? C4 isn't necessarily better, but it's better under certain contexts, certain conditions.
When I was in graduate school, we had a study site. We had gotten a Nature Conservancy grant to do a biodiversity survey at a place called Clymer Meadow, which is down here in the tallgrass prairie. And so there's also an effect of latitude as well as you move from warmer to colder. A tallgrass prairie up here isn't a tallgrass prairie like it is down here. At Clymer Meadow, we were literally like swimming through the grasses because the grasses were way up here, and to get to our study sites, we basically had to create kind of tunnels through the grasses that didn't stay there because the grasses were so thick, and we would just be like walking through the tunnel at night to get to our field sites to collect our specimens.
So think about tall grass as relative to its western counterpart. Here, the tall grasses can be 8 feet high.
Easily.
Here, they're more like, or more. Here, they're usually around 6 maybe, and then up here, it's more mixed. There's a mixture of tall and short grass prairie, tall and short grass species.
Miserable to walk through, by the way. Miserable to walk through.
Okay, so we're going to wrap up with a comparison of a C3 and a C4 plant. We're going to think about some different attributes of these things. Do you remember the common name of this grass species, one of our prairie plants of the day? Big blue stem, correct. Andropogon gerardii, big blue stem. And then over here we've got a salvia. Does anyone know the common name for all salvia species? Or do anyone recognize this one as the common name of this one? Salvias are sages. So this is blue sage. Salvia pitcheri. So this is a 4, do you think it's going to be a C3 or C4 plant? Most 4s are C3. There's a few exceptions, but this one's in the mint family, and as far as I know, all mints are C3. So we've got a C4 plant and a C3 plant, our closed circle and our open circle. As we increase the availability of light, is that increased light availability, which along with it goes higher temperatures, is that going to favor big blue stem or blue sage? Yeah. What are you seeing that gives you that answer here?
I can't see that far, but I know
Oh,
that
okay.
I know that it's a C4 grass and it's probably closing its stomata, so it is not losing as much water that you.
Good. Well, so without seeing the graph from where you are, the other thing we're looking at here is where is the plateau? Where does light saturation start to occur? Where do you see the plateau for Salvia pitcheri here? Someone up closer. Roughly where in the photosynthetic photon flux density axis? Yeah, somewhere around here, 1,000 to 15. It's typically around 1,000 in any C3 plants. Where's the plateau for big blue stem?
Really high.
Yeah, we've gotten there at this point, right? Probably another point would be about here, and then the curve would fit a little better. And as Sonia was saying, it's because they can continue fixing carbon dioxide even with the stomata closed under high temperatures and bright conditions. What about water use efficiency? Is water stress more likely to favor big blue stem or blue sage? Here we've got unburned versus burned, and lowland versus upland. Do you think a lowland or an upland is going to be more water stressed for a plant? If you're a plant, would you rather be growing, and you need a lot of water, would you rather grow in the lowlands or in the uplands up on a hill slope? Yeah, so down here in the lowlands, that's where there's less water stress, and upland's going to be more water stress. So, is water stress more likely to favor Andropogon gerardii or Salvia? And I guess another way to ask this is, which one's going to have higher water use efficiency? Yeah, we're going to have higher water use efficiency with the C4 plants. And notice what happens as you go to a burned upland. What's the water use efficiency there compared to the water use efficiency of burned upland for Salvia? Yeah, much higher water use efficiency, and this one is still photosynthesizing. Whereas what's this one going to do with Stomata in an upland where there's not much water? And they close them, and you get lower photosynthetic rates. What we haven't talked about in this graph is that the gray hash bars are plots where there's more nitrogen available. So these are nitrogen fertilized areas. So let's think for a moment about nitrogen availability. Is low nitrogen availability here going to favor big blue stem or Salvia? Think about this in the context of those resource ratio diagrams. Go back in your memory. Which one is able to persist using less nitrogen per unit biomass? Which one is more efficient with nitrogen use? So there's less nitrogen per unit dry mass in the big blue stem. So it can grow and persist at lower nitrogen levels. So it's got higher nitrogen use efficiency, and if you have plenty of nitrogen, then Salvia does just fine. But if nitrogen is severely limiting, then big blue stem, on average, does better. It's more efficient with the nitrogen use.
Yeah, what this graph is showing is if you took these two plants and ground them up and then measured how much nitrogen is in the plant itself, that's what the graph is showing. So Androphogon has very little nitrogen in it relative to Salvia, so that means that it's probably requiring less nitrogen in order to grow because there ends up being less nitrogen in the plant body.
I appreciate Paul saying that, because even though I keep saying you guys, make sure you read the axes, we also have to make sure we know what they mean. Okay? So yeah, this is the amount of nitrogen that you get.
It took me a few seconds to work through the graph in my own head as well, so.
So you should be able to fill out a table kind of like this. Based on comparing maybe water use efficiency, would you expect it to be higher or lower in C3 versus C4? The light intensity at which photosynthesis saturates, that plateau, is that going to be higher or lower in a C3 or a C4 plant? So I want you guys to think about this, and my plan was to have you talk about it in your group. We have about 5 minutes. So think a moment in your group for the last 5 minutes today. Try to fill this in. We'll make sure that we're all on the same page at the beginning next time, and then we'll move on to the next topic. Yeah.
So take a few minutes to do that.
And if they quiet down, soon enough, then we'll get these back.
They're in alphabetical order, so. Let's see, Savannah.
Okay, well, water use efficiency at C4. What is the one about like?
Kate Hilliard.
Oh my God.
See?
That's a crime.
Laurel.
Optimal temperatures for photosynthesis.
Sydney.
C4 can do it when it's hot.
Nina. David.
Amanda.
Lily Kaiser. Your grades are on the last page, so flip over there, find your grade. Are you growing? Okay, continue discussing photosynthesis.
Right there with you.
It's crying.
Why? What were you saying?
I'm happy. Yeah, I'm happy with it, because I know I failed it.
You need to be a better assessor of your own knowledge.
Yeah. Then you're never disappointed. Then you're never disappointed.
All right, you want to finish the debrief?
Okay, so let's talk about this for a moment before you guys get too into looking over your tests. Water use efficiency, is that going to be higher in C3 or C4 plants? C4. Make sure you remember why. What about risk of photorespiration, higher in C3 or C4? Who has the higher risk of photorespiration under dry conditions? C3, right? Because this one doesn't have that carbon concentrate mechanism. The visco is exposed to oxygen. Light intensity with photosynthesis saturates higher in C3 or C4? I hear both. It's going to be C4. They can continue to photosynthesize it under bright conditions as a C4 plant. Optimal temperature photosynthesis, is it going to be cooler or warmer for each one of these? Which one's going to have a cooler optimal temperature versus a warmer optimal temperature? C3 is going to have a cooler optimal temperature. C4 is going to have a warmer. Good. All right. If you have questions about this, feel free to come talk to us in office hours.